Good morning, my dear students. Today we are going to take up a very beautiful topic for our discussion, and that's what is called as a biotechnology. Now, just like in any engineering course, biology is also developing as a technological course. So, what are all the areas which could be made technological? It's not all the branches of biology. For example, morphology, taxonomy. So they are only traditional subjects. In fact, biology we can classify into two areas: classical biology and modern biology. Classical biology had its origin long back, 3000 years back, when people like Socrates, Theophrastus, Plato, all these people, when they started thinking about the plants, that day the classical botany or the classical biology started. What is the difference between a plant and animal? How the plants could be classified? How the animals could be classified? This is the way in which they were thinking. That is classical biology. But after the period of Johann Gregor Mendel, our attention in biology took a different shape. More or less it was chemistry applied biology, it came. In fact, it was a biochemistry. So people who were working on the different structures in chemistry, they turned their attention to the biological materials. What a plant material is containing, how much of a protein, how much of a fat, how much of a carbohydrate, what are the different vitamins it is containing, and what is the molecular structure of the different hormones that it is containing. So it is in that angle people started thinking in the 19th and the 20th century. So when Watson and Crick gave a model for the DNA, people started thinking in a different angle. When we know for clarity, for when we know very clearly that it is this DNA which is responsible for the determination of a character, why the characters of the organisms cannot be modified by using the genes? It is in that angle they started thinking. If a gene is going to decide and determine a particular expression of a character, why not the character be modified by modifying a gene? Now, when people started thinking like that, then in the field of agriculture, animal husbandry, and in the related areas, people thought that they could improve the quality of the crops. They could improve the milk production in a cow. They could improve the protein production in a fish, the prawn. And like this, people began to think, and then it took a shape more or less of what we call as a biotechnology today. Just like any other subject, biotechnology also has what is called as principles. It has got certain principles, and we have to follow these principles if we want to biotechnologically produce an organism. Set principles are there for this. If you want to improve a crop, if you want to cross two plants, and the best of the two in a single plant, then you have to follow a certain procedure. You have to follow certain principles, and these are all going under a category called biotechnological principles. Then people have evolved different tools for the biotechnology. This is called biotechnology tools. And then finally we are going to what is known as biotechnology applications. In what are all the areas the biotechnology could be successfully applied to derive what we wanted to get. This is what is called the biotechnological applications. So this area of biotechnology, I am going to divide into 4 lectures, 3 coming mostly under the principles and 1 coming under the application. Okay, so there are 3 lectures under the principles. The first one is going to be once again on principles. So biotechnology, principles and process. Before proceeding into the different aspects of biotechnology, first you should understand what a biotechnology is. So I am giving a definition. Definition by European Federation of Biotechnology, EFB. They say that it encompasses both traditional view and modern molecular biotechnology, the integration of natural science and organisms, cell parts thereof, and molecular analogues for product and services. That's more important, for product. Not only for product, also for services. Even if you can derive some benefits for services, it is only biotechnology. So it is an integration. It is an integration of what? It is an integration of a natural science. It's an integration of natural science and organisms and cell parts and thereof to get a product and to get a service. Okay, now I think I have explained it to you very clearly what biotechnology is. It is an integration of natural science and organisms, cell parts, and to get a product as well as a service. So this is what is called as biotechnology. Okay, fine. What it deals about? It deals with the techniques of using living organisms to produce products, as just now I told you, and processes useful to the human. So we get some products by biotechnological processes. So this is the benefits of the biotechnology. What are the products that we are getting? Conventionally, we were making curd, bread, wine, and microbe-oriented process thought as a form of biotechnology. Today, it is restricted to refer to such of those processes which use genetically modified organisms. Very important. So, biotechnology means, in fact, making a curd is also biotechnology. Preparing a bread is also a biotechnology. You are manufacturing a wine, it is also a biotechnology, because you are using a microorganisms there, and then it involves some technological processes. If you just take a cup of milk, pour few drops of a curd, next morning the whole thing becomes a curd. This is also biotechnology, because lactobacillus bacteria are involved there, bacillus bacteria are there, and they do the fermentation. And this could be done in a large scale also. So these are all some of the traditional, conventional methods. So in a very strict sense, this cannot be called as a biotechnology, in a very strict sense. So today, in a very strict sense, what exactly biotechnology means? If you are going to produce a genetically modified organism, then only it is a biotechnology in its a very narrower sense. In a broad sense, it is the production of any product. For example, so many medicines we are producing, so many acids we are producing, so many enzymes we are producing. So all these are also coming under the umbrella of what is called as a biotechnology. So it's a very big area, in fact. Other processes include in vitro fertilization leading to test tube baby, correction of a defective gene, synthesizing a gene and using it, developing a DNA vaccine. Okay, so 4 more areas I am including in the process. One is an in vitro fertilization. You may be wondering why in vitro fertilization, sir, you may be asking. There are so many people who are coming under a category called a childless couple. They are not bestowed with a beautiful baby after the marriage. It's a nature's decision. So for those people, test tube baby is a boon. So they undergo what is known as an in vitro fertilization. That area called a test tube baby, in vitro fertilization, that itself is a very broad area. So many mechanisms are there. In fact, I can tell you that biotechnology is a maximum utilizer only in that area, which is benefiting so many childless couple of this world. And once a baby is born, sometimes they are born with some metabolic error, inborn diseases, congenital defects. So to correct these genes, if they are identified at a correct stage, say at the age at the stage of 6th month or 7th month or 4th month, if there are techniques, we have got the techniques for everything. So by the blotting techniques, we can understand whether a gene is in its correct form or it is a defective form. If you think that it is a defective form and it is affordable to you, you can just modify or get the gene corrected, or the people can go for abortion. If it is going to be a very defective gene and the correction is not possible within their means, so we can take a wise decision in that angle. So defective genes can be identified, corrected if possible, and then synthesizing a gene, you can synthesizing a gene, a correct gene, or a gene which will be useful in agriculture, and developing a DNA vaccine also. So these are all the different areas which biotechnology has flourished well, and then it has made the human life more happier. Escherichia coli, a bacterium found in the bowels, has become one of the potentially most powerful tools in genetic manipulation. Now we have the ability to find specific genes, to cut them away from the chromosomes, to insert them into chromosomes of other species. So there are 3 areas are there, or 3 steps are there. First step is to identify, to find a specific gene. You see, you find a particular gene. So NIF gene, let us say nitrogen fixation gene, or gene which is responsible for fixing the nitrogen which will be in the rhizobium. They are all seen in the root nodules of a leguminous plant. They are able to fix the nitrogen which are in the soil. Now there are so many crops, hundreds of crops are there which are not able to fix the nitrogen. So if this NIF gene could be removed and then inserted in a non-leguminous plant, it becomes a much beneficial to it. So first one is to find a specific gene. You find where that gene is there in rhizobium, then cut them away. So you remove it and then insert it into the chromosome of other species where you want to transport that particular gene to do a particular function. So this is a simplest way to explain the biotechnology. To identify a desirable gene, to remove the gene from its natural environment, and to introduce it in the new environment. So if you are going to do, then you will be deriving some benefits out of it. So this is what is called as biotechnology. Several important species of carrot, cabbage, citrus and potato can be grown from single cells. This is called a tissue culture. See, E. coli bacterium, I was telling you that it is living in the bowels of the human being. It is a peritrichous one. It is containing the flagella throughout its body surface, and from this end to this end, they have already. It is a 3 mu m. 3 mu m. That is what is a 3 mu? You know, it is 1 by 1 millimeter divided by 1000. 1 millimeter divided by 1000 is 1 mu. 1 mu. Okay, microns. 1 mu is equal to 1 millimeter divided by 1000, and so this is a 3 mu in its size. Okay, 3 mu in its size. Now, I want to tell you one very important thing. See, you have to know. Sometimes they have written 3 mu m. 3 mu m. This can be written simply as 3 mu. This and this one and the same. 3 mu m and 3 mu one and the same. Or you can write it even 3 micron. All are one and the same. But remember, if you are going to write 3 m mu, m you are putting in the front, then that becomes different. Remember, this and this are different. This is millimu. This is a mu. This is a mu and this is a millimu. Now 1,000th of this is this one. Okay. So you can, when you are writing a micron, you can put either mu m or a mu or micron. But this is something different. Okay. Now, so 1 millimeter divided by 1000 is called mu, and the size of the bacterium is 3 mu or 3 microns. Now, this is the DNA, this is the cytoplasm. You don't, I have not labeled it as a nucleus. Please remember, our bacteria are all prokaryotic. They don't have a nuclear envelope. So, the naked DNA is present just in the center. It's called as a nucleoid. It's called as a nucleoid. What is a nucleoid? Oid means in Latin which looks like. It looks like a nucleus. It behaves like a nucleus. But it is not a nucleus in all its a true sense. What do you mean by true sense? A nucleus means it will be having an outer nuclear membrane, inner nuclear membrane, perinuclear space, a nuclear pore. All these things should be there. But nothing of that sort is there. Nothing of that sort is there. So it is called as a nucleoid. So it contains a DNA, cytoplasm, cell membrane, flagellum. So these are all the different parts of E. coli bacterium, which is used as the best technological tool. It is giving a helping hand for all the biotechnologies in this world. Yeah, a beautiful microorganism which has been completely, totally, thoroughly worked out for biotechnological purposes. Okay, that's good, fine. Now, anything if you take in this world, it will have its own limitations also. You will have both the positive side and the negative side. You cannot imagine a thing which is having only positive side, positivity. No, nothing is there in this world like that. It will be having certain plus points, certain minus points. Similarly, here also, in biotechnology also, it has got its own limitations. What are the limitations? Once a gene is introduced into a cell, a clone of that cell can produce a countless altered progeny. However, there is a limitation called polygene. There is a limit. Polygene, which is containing 5 or more controlling plant traits, could not be yet identified. Okay, so this is the first limitation. What is the first limitation? See, if you are going to take a cell, if you are taking a gene and then introduce into a cell, a clone of that cell will produce a countless altered progeny. Okay, that's good. But this is not applicable for polygene. When you take a gene, many genes, controlling a plant character, say growth rate, size of the edible parts, and amino acid balance. If you are dealing with these characters, what characters? characters such as a growth rate, size of edible parts, and amino acid balance. If you are dealing with these characters, then it becomes very difficult because these genes cannot be identified. Okay. Further, it is difficult to clone 5 or more genes controlling a trait. These are inevitable disadvantages in recombinant DNA technology. So this is one of the limitations that we are having in biotechnology. Now we are straight away going to the principles of biotechnology. 2 core techniques that enabled the birth of modern biotechnology are genetic engineering and maintenance of sterile ambience. Okay, these are the 2 core techniques. So these are the 2 core techniques. First one is genetic engineering, second one is maintenance of sterile ambience. Now we will go to the first one, genetic engineering. Techniques to alter chemistry of a genetic material, that is a DNA and RNA, to introduce them into host organisms, changing the phenotype of the host organism. This is one level, one core, one technique. So this technique involves genetic altering the DNA and RNA, altering the chemistry of genetic material such as a DNA and RNA. If you are altering the DNA and RNA, changing the phenotype of an organism, now that is the first step. Then maintenance of a sterile ambience. In chemical engineering, processes to enable the growth of only the desired microbes, eukaryotic cell, in large quantities for manufacturing biotechnological products such as antibiotics, vaccines and enzymes. This is the second core of the biotechnology. So in the first one, you are altering the DNA or the RNA or the genetic material, and in the second one, you are producing the growth of the cells. As a result of that, you are able to produce the antibiotics, vaccines and enzymes, etc. Now, asexual reproduction preserves genetic information. Sexual types permits variation. Traditional hybridization in living organisms very often leads to inclusion and multiplication of undesirable genes along with desired genes. See, of course, there is what is known as I was just now discussing, it has got both the minus and plus points. Normally, you say that in nature favors only sexual reproduction because the viability will be more, and so many advantages are there in the sexual reproduction. But one aspect of sexual reproduction is when you take an organism and then this is another organism, and then when you mix these 2 organisms, organism A and B, then what will happen? The character will get mixed. The character is getting, this is a male and then this is a female. The character is getting mixed. You get a mixed population. But the other one, if an organism is A and if it is asexually reproducing, then a due replica of this one will be produced. All the individuals produced from that will be having all the same characters of the A. So, asexual reproduction preserves the genetic information. It is not getting contaminated. It is not getting mixed. It is not getting altered. It preserves the genetic information asexual. So in that angle it is good. Viewing that angle it is good. But sexual type, it is permitting the variation. In a traditional hybridization, what happened in living organisms very often leads to inclusion and multiplication of undesirable genes entering. Undesirable genes entering. So this is where we are using the biotechnology. If you are hybridizing a plant, two plants, then undesirable genes will be there. But in biotechnology, that can be eliminated. That can be eliminated. I think I can explain this in another way also. See, you are taking plant A and then plant B. Now, in a traditional hybridization I am talking, this is having a small fruit. The fruit is small. But at the same time, it is very tasty. So very tasty. Plant B is having a large fruit of the same variety. Say apple or orange or a mango, whatever it is. But at the same time, it is very sour. Now, my idea is to get a large fruit and at the same time tastier. So this is my idea. I want the fruit, the mango fruit, to be larger and at the same time tasty. But I should not forget that an undesirable gene like this, these two may also go together. There is a danger of me producing a crop which is a small as well as a sour. So undesirable genes are coming here. But whereas in biotechnology, this problem is solved. That's what I want to make it very clear here. So undesirable gene along with the desired genes will enter. But in genetic engineering, this problem could be solved. But in genetic engineering techniques, including creation of a recombinant DNA, use of a gene cloning and a gene transfer, overcome this limitation and allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. You are able to remove the genes which are responsible for the sweetness and then introduce into a plant which is already having a large fruit, then the problem is solved. Here there is no question of the sour and the smallness are going together. Are able to follow. So some of the problems that we had in the traditional agriculture is now solved. See, is Paramecium undergoing the sexual reproduction and asexual reproduction. In asexual reproduction, perhaps I will start from this a single organism, and then it is becoming binary fission is taking place. It is becoming mostly cleaved into two, and then now it has become two Paramecium, two, a single. So there is no variation, genetic variation is not there. This Paramecium from which we started this one, all the three are genetically same. All the three are genetically. These two daughters are same, similar. And both of them, they look like their mother. So genetic variation is not there. But if you take this one, you take two, male and the female, and then when they undergo sexual reproduction, there is an exchange. See, there is an exchange of material, and then the gene of one is getting contaminated with that of another by the sexual reproduction. Most likely, this DNA piece transferred into an alien organism would not be able to multiply itself in the progeny of cells of the target organisms. But when this is integrated into genome of the recipient, it may be multiplied and be inherited along with the host DNA, because alien piece of DNA becomes a part of the chromosome which has got ability to replicate. It's a beautiful concept. See, most likely the DNA piece transferred into an alien organism would not be able to multiply itself in the progeny. See, this is an organism. Okay, and then you have got the chromonucleus. Now you are introducing a DNA from a particular organism. Now, if this is not getting incorporated into the original DNA of that organism, then there is no use. See, this is, let us say, this is an organism B, and this DNA is from organism A. Then, if this fragments of a DNA derived from the organism A is not incorporated into the B, then there is no use. Okay. So, but when this is integrated into the genome of the recipient, then it becomes useful. Then it can be multiplied and inherited along with the host DNA. Okay. See how beautifully it is shown. This is the bacterium. This is a bacterium, and this bacterium, this is the bacterial chromosome. This is the bacterial chromosome. This is the recombinant DNA molecule. Okay, as a DNA recombinant molecule. Then this is introduced here. This is the one as a recombinant portion. It is already inserted into the plasmid, and then that is once again transferred here. So insertion of rDNA into the bacteria for replication. A specific DNA sequence is a chromosome in a chromosome is responsible for initiating replication. Any alien piece of a DNA in an organism needs to be a part of a chromosome which has a specific sequence, origin of replication. Because of its linking with origin of replication, alien piece of DNA can replicate and multiply itself in the host. This can be called as a cloning or making multiple identical copies of any template DNA. So, a specific DNA sequence in a chromosome is responsible for initiating the replication. This point. So, this is a DNA. Now, this DNA should replicate. Replication should start. Replication means a duplication. First, for replication, the first step number 1 is unwinding. When it is unwinding, so the place where the unwinding starts, the place where the application starts, it is the point. That point is very important. See, a specific DNA sequence in a chromosome is responsible. So a particular DNA sequence is there that is responsible for initiating the replication. If a replication has to start from a particular point, a specific DNA is very, very important. Any alien piece of a DNA in an organism needs to be a part of the chromosome which has a specific sequence because of its linking with the origin of a replication. Alien piece of a DNA can replicate and multiply itself in the host. This can be called as a cloning or a multiplying or making multiple copies, identical copies of any template DNA. So, how to produce, how to construct, make a recombinant DNA molecule, that is what is called the rDNA. It emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid, that is a autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium. So, this is a Salmonella typhimurium. In this, you have got the normal DNA and then a plasmid. Okay. It emerged from the possibility of linking a gene encoding for antibiotic resistance with the native plasmid. This is the native plasmid. Now, in this native plasmid, what I am going to do? I am going to link a gene. So, linking a gene in a native plasmid in the case of a Salmonella typhimurium. So this rDNA technology emerged because of the first one. It started there only. Stanley Coher and Herbert Boyer in 1972 isolated the antibiotic resistant gene. See? This is a normal DNA, plasmidal DNA. Sorry, I have drawn it very big so that you can see it very clearly. So this is the antibiotic resistant area. So antibiotic resistant gene in the cutting a piece of a DNA from the plasmid which was responsible for conferring the antibiotic resistance. So this portion was removed. This portion of from the Salmonella typhimurium was removed, and then the cutting of DNA at specific locations was made possible with the discovery of so-called molecular scissors. So how can you cut DNA molecules? See, DNA molecules are very small, extremely smaller, so small in their nature that you can't use your normal scissors for excising it. So there are what are called as a molecular scissors are available. These molecular scissors, when added to the DNA or to the bacteria, they find out the origin of origin, and then they go and then they make a cut there. So this cutting of a DNA at a specific location was made possible with the discovery of so-called molecular. There's a molecular scissors called restriction enzymes, which are of two types, which we will be discussing later on, or called endonucleosis and exonucleosis. So one which will be opening or DNA from the end, another one in the middle. So they are the molecular scissors. See, Salmonella typhimurium, it is it's a highly food poison is responsible. Salmonella infections are zoonotic disease measuring. It can attack anyone who eats infected food, and it can be even fatal. Simply, it could be found in high risk foods. High risk food means a food that is highly contaminated. See, even 150 people, any number of people could be affected. So it causes a diarrhea, abdominal cramps, vomit, fever, etc. So the result will be even death. Cut piece of a DNA was then linked with plasmid DNA, which acts as a vector similar to the mosquito is transferring the malarial parasite into a human body to transfer the DNA piece attached to it. Plasmid is used to deliver an alien DNA piece into the host organism. Linking of antibiotic resistant gene with the plasmid vector was possible with DNA ligase enzyme, which acts on cut DNA molecule and joins their ends. That is what is called a paste molecule. See, cut piece of a DNA was then linked with plasmid DNA, which acts as a vector. See, first this DNA is a cut piece. What is this DNA which was a cut? Antibiotic resistance. This antibiotic resistance was cut because it is that gene we want more. So that gene is a cut, and then it was linked with a plasmidal DNA, which acts as a vector. The moment you attach it, and it can be transferred to anything else to transfer the DNA piece attached to it. So this plasmid is used to deliver the alien DNA piece into the host organisms. Linking of antibiotic resistant gene with the plasmid vector was possible with DNA ligases. Just like molecular scissors are there for exonuclease and endonucleases, you have got DNA ligase also. This makes a new combination of a circular autonomously replicating DNA created in vitro and known as the recombinant DNA. So this DNA is called as a recombinant DNA. Now you understand what a recombinant DNA is? See, this is the DNA. This is the DNA, a portion of the DNA, and now you have made a cut and then added a new DNA portion to this. A new DNA has come to this. Now this is what is called as a recombinant DNA. So if I am going to put it in a circular form, if this is the DNA, a cut is made and then you add the new one, then now this has become a recombinant DNA. So this makes a new combination of a circular autonomously replicating DNA created in vitro known as the recombinant DNA. This DNA is transferred into Escherichia coli, a bacterium closely related to Salmonella typhimurium. Because when you are transferring the DNA, you should see that they are all very similar organisms. Okay, then only it becomes easier, which could replicate using new host. DNA polymerases enzyme and make multiple copies. Ability to multiply copies of the antibiotic resistance gene in E. coli was called cloning of antibiotic resistant gene in E. coli. Okay, so cloning of antibiotic resistant gene in E. coli was made possible because first, what do you do here? You identify a gene. What gene you are identifying? Antibiotic resistant gene. Where that gene is there? It is in Salmonella. So you cut the gene, antibiotic resistant gene from the Salmonella, and then you put it in along in a vector, transfer the vector, and then finally it has been now transferred to Escherichia coli. Okay. See how it is a recombinant vector was inserted. The original DNA, was cut and then a portion was inserted. Okay. And now when this is the bacterial genome, when this was inserted, it has become a part of the bacterial genome, and then it starts reproducing, replicating. This is a clone of donor fragments. So the fragment which came from outside, alien gene, that also started replicating. Okay. Now, what are the three basic steps which are involved in genetically modifying an organism? What are the three basic steps? There are identification of a DNA with a desirable genes, introduction of these identified genes into the host, and then introduce the DNA maintenance in a host and the transfer of a DNA into its progeny. So these are the three steps. First one, identification, introduction of this identified gene into the host, and then the maintenance, DNA maintenance by replication, by producing a number of progeny. See, restriction enzyme, recognition sites. Now this site is what is called as a GAATTC. GAATTC. You will be having this just like the word Malayalam. If you read from here, it is called this way. If it is read from here also, the same way. Okay. Now they like this. This place from 5' to 3' and 3' to 5'. In this position, a restriction enzyme cuts the DNA. Okay. So G is left here, other goes here. G is left here and other comes here. Now this becomes a sticky end. This becomes a sticky end. Now in this sticky end, any new DNA could be added. See, DNA fragment produced by the same restriction enzymes, and it gets inserted. DNA ligase is the one which is sealing, or which is combining, which is pasting the new DNA molecule into the old DNA where the cut was made. Events of recombinant DNA technology. What are the different events are there? Isolation of a DNA of donor or gene of interest and cut into fragments using restriction endonucleases. So first point is isolation. Point 1 is isolation. So it is the isolation of a DNA donor. A particular donor is ready for donating its gene. Perhaps it is a useful gene. It is a very useful gene. So you have to isolate that gene from the other genes of that area of the chromosome. Okay. So first one will be the isolation of the DNA in a particular gene which is of interest. Perhaps you want to produce more milk in your cow, and some you want to increase the protein. So for all these things where the gene is present, you try to identify. After identifying is the first step in the DNA technology, and cut into fragments using the restriction endonuclease. So after the isolation, you cut it into fragments. So these two are there in the first step. Then attachment of the DNA into a suitable replicon or a cloning vehicle for plasmid. So this is the third one. First one isolation, second one is cutting, third one is attaching. Now cutting of a DNA vector into fragments using the endonucleases. So the vector also you have to make a cut. Then joining of a DNA fragments of a donor and vector through the splicing, resulting in hybrid DNA, that is what is called as a rDNA, recombinant DNA or rDNA. Then next, what are the next step? Introduction of rDNA into a host cells, that is E. coli, Bacillus subtilis, then Streptomyces, etc. You can put it anywhere. After treating with cellulose, making host cell wall permissible to the entry of the hybrid DNA. So how to put it? You have to first treating with the cellulose, making the host cell wall permeable to the entry of the hybrid DNA. That's very important. You can't simply introduce the rDNA into a host. Multiplication of a host cells with a foreign DNA following instruction of a foreign DNA, resulting in a colony of bacteria having the restriction DNA or DNA. Then last one is what is called as a growing of a colony separately to obtain multiplication of rDNA fragments, resulting in a number of colonies having identical copies of rDNA fragments. See, once a colony is a producer, all the members of the colony will be exactly identical because it is just taking place by the cell division. Okay. Now a typical example we can take the production of insulin from the E. coli. Now what's happened? See, some of the parameters or some of the informations if you hear about this diabetes and insulin are very interesting. They say in India 1/3 of the population is suffering from diabetes. That is, if there are 100 people, 30% of the people, 33% approximately, they are suffering from the disease. Very big, a number. You may think it's only a 30% or 1/3. See, that is a very big. See, in a population of a country like India, where the present population is 110 crores or 115 crores, roughly 40 crores of people are suffering from a disease. 40 crores suffering from a disease. And where they will go for the medicines or for the remedies, all these people. Okay. So they are depending on the insulin. So this insulin was once upon a time got from the animals. The animal the pancreas was removed from a pig, sheep and different animals. It was ground or grinded, and then they purified, then the insulin was taken. Once the people come to understand that it was the insulin that was taken from the pigs and the different animals, some people may not like to take it. So it had some ethical issues also. They had a lot of problems. So, and then we were also not able to meet the demand. The demand was very high. See, 40 crores of people in India affected by a disease, and then supplying the insulin to them is not an easy joke. Are you able to follow? So nowadays what we are doing, we are able to produce the insulin from E. coli. So what we have done? We have identified the insulin producing genes, where that gene is there present in the human being, that we are able to remove, excise, and then put it in the E. coli bacterium, and then these bacteria are cultured in a large scale, and then now these E. coli are producing the insulin. From them we are harvesting, and then we are taking that. So production of insulin from E. coli with restriction DNA. By introducing the gene responsible for the production of a human insulin from the pancreatic cells into a recipient E. coli cell. E. coli is made to produce the human insulin. So what we have done? We have introduced a gene responsible for the production of human insulin from the pancreatic cells into the E. coli, and E. coli is now preparing or manufacturing what is called as insulin for us. Okay, see the human pancreas cell, this is a human pancreas cell, and this is the DNA which is responsible for production of the insulin. Human pancreatic cell DNA is responsible for the DNA which is responsible for insulin secretion. Okay, now this gene is taken, and then it is introduced, then a bacterium, E. coli bacterium is taken, and in the plasmid DNA this gene was introduced. So now you have got a recombinant bacterium, E. coli bacterium, which is recombinant, and now you are able to produce, multiply it millions and millions of in a fermenter or in a tank. You are able to produce this, and then finally the insulin which is needed for the human being is synthesized. So the insulin which was synthesized in the human system once upon a time, when it fails due to the, you may be asking what is the reason for the failure of insulin secretion in our body. I am going to give you 101 reasons for that. The change of lifestyle, change of food habit, so many reasons are there. The time is changing, and many people are turning out to be diabetic patients. So you have to depend on the other organisms to produce insulin for you. The stage has come like that. So these bacteria, these insulins, the genes responsible for synthesizing the insulin is taken, and then it is introduced into the E. coli, and then they are synthesizing the insulin for us, and then we are happy about that nowadays. So these are all I was just narrating to you very understandable way. I think that how biotechnology could make the human life more happier. What are the different principles? What are the different processes involved in that? So the basic principle is you identify a gene, a desirable gene, and then transfer that gene into another organism by means of a vector. From in that, so many technologies are there. You have to make a molecular scissors are there, and once you have to cut it, you have to paste it, cut and paste. In computer we are using that. So you have to cut and then you have to paste, and then you get this modified DNA or called rDNA, and that rDNA is able to do the work that you want to do. A very simple thing. Okay, so this is one angle of biotechnology, and in a series of lectures, as I was telling you, some more interesting information that we are going to have in the next lecture, which is going to follow in this DNA technology, biotechnology. Thank you very much for listening to me, my dear children. If you have got any queries, if you have got any doubts, I am always happy to hear from you. Thank you very much. Fine.